Hello, welcome to Study Hub Pro. Today, we're diving deep into a hidden world that's all around us, and even inside us. We're talking about microbial genetics and molecular biology, basically the study of the tiny engines that drive life itself. We're going to break down exactly how they work, one step at a time.
So, let's start with the big question. What's the real connection here? How does studying the tiny instruction manual of a single bacterium lead to medicines that can literally save millions of lives? That's the core puzzle we're going to unpack in this explainer.
To get to that answer, we've got to start with the basics. So, first up, what exactly is microbial genetics?
At its heart, it's the study of the rulebook for the smallest living things on the planet. We're looking at their genes, how those genes can change and create variety, and how they pass those traits down from one generation to the next.
And this is why it's so important. By studying their genetics, we get this incredible window into how these microbes work, how they build themselves, how they make copies, and how their DNA acts as both a library full of information and a 3D printer, creating everything they need to survive and evolve.
So let's zoom in on that code. You can think of any living thing really as a super complex building. Well, its genetic material is the master blueprint that has every single instruction needed to build it.
This blueprint has a name. It's called the genome. It's the entire library of genetic instructions for that organism, containing every single gene it needs to build and operate itself.
Now, what's really wild is how simple the alphabet is for this code. The entire genetic language is written with just a few letters, called nucleotides. You've got A, C, and G in both DNA and RNA, but T is only found in DNA, while U takes its place in RNA. That's it. Those are the fundamental building blocks.
But, what happens if there's a mistake when you're copying that blueprint? Well, that's what we call a mutation. It's a permanent change in the DNA, like a typo in that instruction manual, and it can totally change an organism's traits.
But for microbes, typos aren't the only way the blueprint can change. This is where it gets really interesting. Microbes can actually trade useful genes with each other on these little circular pieces of DNA called plasmids. This whole process is called horizontal gene transfer, and it's like a massive, super fast genetic file-sharing network. So, with this incredibly dynamic blueprint, how does a cell actually read the instructions and build what it needs?
The answer lies in this really fundamental principle called the central dogma of molecular biology. It's the master process, the main workflow, that describes how genetic information flows from its permanent storage in DNA to a temporary message in RNA, and finally into a functional product, a protein.
And it's really a two-step process. In transcription, a piece of the DNA is copied into a matching RNA molecule. Then, in translation, the cell's machinery reads that RNA message and uses it as a guide to build a specific protein. From code to creation. But bacteria, they are ruthlessly efficient. They use this brilliant genetic switchboard, called an operon, to turn genes on or off only when they're actually needed. No wasted energy.
Okay, so we've seen how proteins get built, but what are they and all the other molecules they work with actually made of? Let's take a look at the essential building blocks of life.
It turns out that pretty much all life, from a microbe to a blue whale, relies on the same four big families of molecules. You have carbohydrates for energy, lipids to build membranes and barriers, proteins to do all the work in the cell, and of course, nucleic acids like DNA and RNA to hold the master plan.
Together, these four groups make up most of a cell's weight and are the absolute foundation for everything it does, from giving it its shape, to powering all of its activities, to storing and passing on its genetic legacy.
All right, we've seen the blueprints and we've seen the building materials. Now let's take a peek inside the workshop where all of this actually happens, the cell itself.
This brings us to another one of those fundamental concepts, the cell theory. It's super simple but profound. It just states that the cell is the most basic unit of life. Every single living thing is made of one or more cells, period. And these cells come in two main flavors. On one hand, you have prokaryotic cells. They're simpler, they don't have a nucleus, bacteria are the classic example. On the other hand, you have eukaryotic cells, which are much more complex with lots of internal compartments. They make up plants, animals, and yep, us.
Okay, let's connect all these pieces back to that original question about medicine. Because once we understood the rules, the blueprint, the gene swapping, those on-off switches, we had a huge realization. We could use these rules ourselves.
This is the whole basis of recombined DNA technology. It's like a toolkit for cutting and pasting genes to create powerful new solutions.
Now, remember that idea of a mutation we talked about? That typo in the blueprint? This is where that concept becomes incredibly important for human health.
So here's the chain of events. It's a direct line. A mutation happens in a gene. That means the protein it's supposed to build gets made incorrectly or misfolded. That misfolded protein might not work at all. And when a protein that does a critical job fails, a disease can develop.
And this isn't just some abstract idea. This exact process is the root cause of many well-known genetic diseases, like cystic fibrosis and sickle cell anemia. It also plays a major role in many types of cancer.
And this is the punchline. Understanding the blueprint doesn't just let us diagnose diseases, it empowers us to design cures. Using that recombinant DNA technology, we can actually engineer bacteria to produce life-saving insulin, or we can develop targeted therapies that go in and fix the problem right at its molecular source.
This whole journey leaves us with one final, really powerful question. We've learned how to read the code of life. We're getting better at it every day. So what's next? If we can read the blueprint, what's stopping us from rewriting it?
Now that's a topic for another time. See you in the next video.